Okay, so we're going to begin looking at Chapter 2. Principles and Concepts, Parasite Systematics, Ecology and Evolution. This section will cover systematics and taxonomy of parasites. We have to keep in mind the invertebrates, whether we're talking, the multicellular parasites, the single-cell protozoa, the worms, the arachnids, whatever, insects, all of the above. They're not as well known, they're not as well studied, they're not as well identified as us verts. So because of this, as we start to look closer, as we start looking and poking around in all the nooks and crannies on this planet, every year we're finding and we're identifying new species of invertebrates. And what's kind of cool about that is if you find something, you get to name it, which is kind of cool and kind of off the wall at the same time. So, it's led to some issues. People find something and they want to name it. Generally it's either named after the person who found it, they Latinize their name or, turn, some form of where it was found. Or, as, the past decade or so we've seen, they name it after celebrities or they name it after their dog or something like that. And when it comes to the invertebrates, the parasites, we follow the normal taxa, okay? Grouping like with like. Start off with the big umbrella, and you start going smaller and smaller and smaller in your groups until you hit the genus and the specific species, okay? So we follow, in microbiology and parasitology, we follow the normal Linnaean means of grouping like with like, kingdom, order, phylum, blah, blah, blah. And yes, I've got those out of order because it's been so long since I've thought that way. And but what's interesting is we're constantly updating, we're constantly changing this taxonomy. We think we may have found an orphan species all on its own. It's the one and only of the genus. But as we start studying it more, we find that no, it's not an orphan species. It belongs to this genus, which in turn belongs to this, belongs to that, and so on and so forth. So we're constantly redesigning the relationships. And a lot of this is due to our modern take on systematics. You go back 4 or 5 decades ago, systematics, the phylogenies were based on physical appearance, where they are found, and maybe some of the biochemical aspects. What abilities did that organism have that are similar to other organisms? But now what we've started doing is we've started collecting DNA sequencing. DNA sequencing is now relatively inexpensive. I mean, it's still expensive. You and I can't afford to just go and pay to have a complete organism completely, have its genome completely sequenced out of pocket. Well, you could. Daddy Warbucks money. If you got Daddy Warbucks money, you can go and do this. It's still going to cost a couple of grand. You go and one of these sequencing labs, it'll take them a few weeks, but then they will send you a file with every A, G, C, T in the DNA. Give you a complete sequence. You can then, that sequencing nowadays isn't that hard. It's the going through and characterizing. It's going through and determining what's a gene, what's not a gene. On the gene, what's the exon, what's the introns. That's where a lot of the computing power comes into play. And that's where it takes a lot of time and a lot of expense. But as we begin taking these DNA sequences and putting them into GenBank, and we start seeing what it takes to get more and more of them together, we can start comparing. I mean, going onto PubMed, type in different genus species, and you can get, if they've had anything uploaded to GenBank, you can get it. And with a couple of clicks of a mouse, you can start comparing DNA sequences, finding similarities, finding differences. You can import it into other programs that are readily online, and it can start giving you, best guess as to, what is the introns and exons, what are the gene sequences in this long section of DNA that you've uploaded. So because of that, we can now take and we can now start comparing A to B, A to B, C, and D, and seeing how they relate. No longer just based on physical appearance and biochemical, we can start comparing DNA. We can go through and start comparing the DNA sequences. We know that, there's going to be some mutations at a given rate over a given amount of time. That's how we can sit there and compare DNA lineages in humans. We know within the short lifespan that we've had, Homo sapiens, there's going to be so much mutation. It's going to be separating us, out. Well, we can take that same theory and then we can just apply it to the bigger time frame that we would think of as evolution that's going to be working under. So we can start comparing and trying to determine, based on mutation rates and things like that, speciation rates, organism A is more closely related to B, which is B, A and B are more closely related to E than they are to C and D, and we can start coming up with our own phylogenetic trees. We can also do this with ribosomes. Ribosomes the genes for those are relatively stable. Anytime you get a mutation in the ribosome sequence, the ribosomal RNA is not going to be the correct shape, there's no function, which means that organism's not going to develop beyond the zygote because it's not going to be able to make proteins, the polypeptides, at the needed rate. So ribosomal subunits are going to be relatively stable genetically. Okay, yes, there's still mutations and all, but it's not going to be at a high rate. So we can start comparing ribosomal sequences. See whose sequence is more closely related to who, so that way we can start comparing, okay, these would have had, since they have the same changes, relatively the same changes between these two species, these two genuses, we can start to sit there and say, okay, these are more closely related than these ones. We're moving beyond total genome, ribosomal RNA, and now looking more at picking one gene that we know is going to be across all eukaryotes. One gene that's going to be there, and this is the one we've chosen, cytochrome C oxidase, specifically the subunit 1, CO1 is what the acronym for that one is. We are looking at, not only the DNA sequence, we're looking at any intron-exon structures, looking at the overall polypeptide itself, and we are comparing like with like to dissimilar, and we're sitting there and trying to group, relative groupings, who's closer to who. And this is where we're at right now, systematic-wise. As we go through, more information comes out. As we go through some of the older, been characterized and put in a database many, decades ago, and we start pulling them out, and we're, doing the sequencing. So it's not just the new stuff. We're now also looking back, things that have been cataloged. Samples put on ice, frozen away. Well, let's dig those out. Let's get some DNA. Let's start looking at them so we can now get this bigger database, DNA database of, total genome, RNA, and now cytochrome C, which is the big push. So we can start getting a big, broad idea of the relationship between organisms. Who came from whom? What was the origin of that lineage and things like that.